let us start our oogenesis. As we discussed about gametogenesis in female oogenesis and male spermatogenesis. So first let us start with oogenesis. It is formation of mature ovum from the oogonia. So we're having here that is prenatal development, also postnatal development. So prenatal development is that is taking place before the female is born, and postnatal development taking place after the female is born. So let us start with prenatal. So the ovaries start oogenesis in a prenatal life only, that is the primordial germ cell appear around the 4th week of a yolk sac in a wall, like proliferating mainly by mitosis process. So this migrate towards the gonads of a developing female. So here what happens is this primordial germ cell undergoes mitosis and forms oogonia. So oogonia also undergoing mitosis inside and forming a follicular cells around the oogonia, and this is known as primary oocyte. So here the genetic material is same throughout until the primary oocyte, that is it is diploid in condition and having two-end structure, that is the chromosome over here is double structured. And next the primary oocyte is actually arrested at the prophase, and here even it is diploid, but the it is double structured here, that is 4-end, and it is mainly struck at the prophase of a diplotin stage. So what happens, let us continue the story. So the primordial cell, oogonia, primary oocyte, and ready for the meiosis. So at the birth what happens is the primary oocyte are present in infant and they are arrested in the diplotin state of prophase. At the 5 months oogonia are about 5 million, and the primary oocyte have flat epithelium cells around it, that is known as follicular cells around them, and this is also known as primordial follicle, remember that. So now let us understand what happens after birth. It is postnatal development. So after birth up to puberty, still the primary oocyte is arrested in a prophase, that is an diplotin state prophase. But at the puberty what happens, because of follicular stimulating hormone, it stimulates growing, that is 5 to 15 primordial follicles start growing, and the follicular cells become stratified cuboidal cells, multi-layered and forming a primary follicle. So now what happens, the primordial follicle is converting into growing follicle and growing follicle is converting into primary follicle. But still remember that the primary follicle is still having a primary oocyte inside. So this primary oocyte secretes the glycoproteins between mainly a oocyte and a follicle, forming a zona pellucida. So the fluid filled space, the antrum and some follicular cells remain called as cumulus oophorus. With the connective tissue, they form the thickness around the follicle uh, like a follicular, and this is forming a secondary follicle. So secondary follicle is also still having the primary oocyte. So the primary follicle having still primary oocyte, and even the secondary follicle is still having the primary oocyte. So the theca folliculae, that is internally they form the theca interna and outer they form the theca externa. So here connective tissue is nothing but forming this theca folliculae. So interna, theca interna, outer theca externa, both are forming mainly estrogen in the female. So the primary oocyte now complete the first meiotic division and form the secondary oocyte now, along with one polar body, and between this zona pellucida and the secondary oocyte, um, this is present. So it is having 23 haploid and 2, and that is double structure. And now this secondary oocyte undergoes second meiotic division, that is fully matured. This is fully matured secondary follicle is known as Graphene Follicle. So whenever there is LSH, that is ovulation takes place 3 hours before ovulation, the secondary oocyte also start the secondary meiosis and arrest in the metaphase. So now what happens when ovum along with the cumulus oophorus taken up by fallopian tube along with the polar body, and the remaining follicle remain as a corpus luteum, which is yellow, produced by and also produce progesterone. And now the cells of the cumulus arranged as corona radiata. So if the sperm doesn't come, the ovum wait for about 24 to 48 hours. Later they disintegrate and die in the fallopian tube. So if the sperm comes, the magic tug of sperm, what it does of the cell membrane, the secondary oocyte undergoes second meiotic division and produce one more polar body, that is produce one polar body. First polar body produces 3-4 polar bodies. So the definitive ovum is formed finally, and it is fertilized ovum, so its life is like up to full human body. So if the ovum is not fertilized, it may die for 48 hours, but if a ovum is fertilized, it forms a full human body, right? So the definitive ovum is haploid, and it is single structured chromosome. And now the female and male pronucleus together bind and form a zygote, which is diploid 46 chromosome. And we know that if from male it is X or Y. If X, then female is born, because both sides X in case of female. If Y, then male is formed. So this is just about oogenesis. Next, let us continue about male spermatogenesis, which is very simple than this. Before discussing spermatogenesis, we'll also discuss the diagrams over here, so that it will be like a revision. So the number of chromosomes in somatic cells of man and woman, you can see in a man 44 X and Y, right? Autosomes are 44, X chromosome is 1, Y chromosome is 1. In the case of woman, you can say again 44 plus both X. So the autosomes are 44, X chromosome is 2, and the Y chromosome is 0 in the case of women. So here let's see the diagram to show the parts of typical chromosome. So each one is a chromatid, right? So both chromatids are bind at the centromere, and this is the long arm of a chromatid. This is the secondary construction satellite cells, and this is a short arm of chromatid. So this is like a very fast revision of all the diagrams which are present in our textbook. So you can see this is a scheme to show the main stage of meiosis. So we know meiosis, so it's not necessary to revise. Okay, let us revise. So this is the early interphase. Chromosomes are in the form of extended threads, and here you can see the late interphase. The DNA of each chromosome has undergone duplication now, and here you can see say, and see is early prophase. Chromosomes now seen distinctly, centrioles move apart, and here this is late prophase. Nuclear membrane disappears, spindle forms. Metaphase, chromosomes arranged at the equator attached to the spindle. Anaphase, centromere split, so that each chromatid becomes an independent chromosome. One chromosome of each pair moves to either pole. And telophase, nuclear membranes are formed again, centrioles divide, chromosomes gradually lengthen and become indexed. So as we are discussing it faster, because we already know the short process, just to complete the diagrams. So here we know the cell cycle, 4 phases: G1, pre-synthetic, S phase, G2, M phase. Yeah, so this all we know already. Yeah, and here we have that M phase, in which the cell divides into two daughter cells, and sister chromatids split at centromere and move into two daughter cells, right? So this is the stages in the prophase of the first meiotic. This is meiosis 1. So the chromosomes become visible. Each chromosome is made up of two chromatids, but these cannot be made out separately. Two homologous chromosomes come to lie side by side, forming bivalent. Four chromosomes distinct and form tetrad. Two central chromatids become coiled on each other so that they cross at the number of places. Only one such crossing is shown, and the chromosomes now separate. The central chromatids break at the points of crossing and unite with the opposite chromatid. So we discussed, so just fast revision. So metaphase, the nuclear membrane disappears, spindle forms, chromosomes are attached to it by their centromeres. And anaphase, on entire chromosome, one pair moves to either pole. Note that centromeres do not divide. Telophase, note that chromosomes each cell have been reduced to haploid number. Yeah, we know this. So this is daughter cell resulting in second meiotic division. These are not alike because of crossing over during the first meiotic division, right? So this is like fast. Next, so gametogenesis we are now in the main process. So this is the parts of the spermatozoon, like this is the acrosome, this is the head, this is the neck, middle piece, annulus, principal piece, tail, and piece. So remember that head is divided into centile and basal body also. So parts of the spermatozoon in somewhat detail here. Check head, neck, middle piece, principal piece. This is about 10 times as long as middle piece, right? And you can see this Golgi apparatus forms the acrosomal cap, nucleus for the head. Mitochondria form the sheet of the middle piece, and proximal centriole comes to lie in the neck. Distal centriole becomes ring-shaped and forms the annulus. Axial filament grows out from the centriole. It is present in the middle piece and also in the tail. So this is a transfer section across the principal piece tail of a spermatozoon to show arrangement of fibrils. So the coarse petal, fibrous sheet, doublets, plasma membrane. So it's like cilia or microvilli like arrangement, right? Microscope is structure of seminiferous tubule. We already seen histology. So now let us learn about spermatogenesis as we completed oogenesis. So this is important now. So same like meiosis process, it continues. The spermatogonium first of type A. Type A nothing but it undergoes mitosis and just remains there. But the type B is important. So type A undergoes again mitosis. One of it may degenerate or go anywhere, but one of it forms spermatogonium type B. So this type B plays important role in case of forming a spermatozoan. So it is X and Y with 44 homologous. So it undergoes mitosis. That is spermatogenesis from about 1 million sperms. So mitosis, here primary spermatocyte is formed. So spermatogonium into cyte now. And the spermatocyte is diploid and it is only single structure. So we undergo first meiotic division starts here and forming the secondary spermatocytes. So primary spermatocyte, secondary spermatocyte. Remember, secondary spermatocyte as it is meiosis is reductional division, so then they become haploid, but they are double structured, right? So they undergo secondary meiotic division forming 22XX and 22YY here. So here it is completely haploid and single structured chromosome, right? So these are now called as spermatids. Now these spermatids are transformed into spermatozoa. So spermiogenesis, this process is called spermiogenesis. So whole process is called spermatogenesis, but only conversion of spermatid into zoa is called spermiogenesis. So first you can see the order like first gonium type B. After gonium we have our cyte, primary spermatocyte, I mean spermatogonium type B, next into primary spermatocyte, next into secondary spermatocyte, next into 4 spermatids, after spermatids spermatozoa, 4 spermatozoa. So this is how they are formed. This is very simple process of spermatogenesis and also spermiogenesis means spermatids into spermatozoa. So very simple process. Now stages in spermiogenesis like meiogenesis means from spermatid to spermatozoa. So first one is Golgi complex is formed, nucleus, mitochondria, centrioles are formed, cell membrane is formed, axial filament is formed, and you can see or axosome head, proximal centriole neck is formed, middle piece, mitochondrial sheath, annulus, distal centriole, and middle piece and tail axial filament, membrane covering sperm. So with this we know panoramic view structure of ovary. We already know about ovary a lot. We discussed. So this is stages of oogenesis. Oogonium forms primary oocyte. Primary oocyte undergo first meiotic division form first polar body. Secondary oocyte forms second polar body. Second meiotic division to form finally ovum. So these conditions we already know in the meiosis. So you can check out this is oocyte, primary oocyte, nucleus, flattened and follicular cells, stroma. So this is zona pellucidum, primary oocyte, primordial follicle, follicular cells become columnar basement membrane. Absent zona pellucida only in follicular cells. Okay, early stage in formation of ovarian follicle. So the formation of membranous granulosa. Follicular cells form membranous granulosa, primary oocyte, zona pellucida. This we'll see in detail in our menstrual cycles and female fertilization, etc. So formation of follicular cavity. There is this antrum follicular cavity. This is primary oocyte, right? So this is a fully formed ovarian follicle, that is a Graphene Follicle, mature follicle. So we have theca externa, theca interna forming estrogen and membranous granulosa, and this is secondary oocyte here. Remember, right? In Graphene Follicle we'll have secondary oocyte, right? Here we have cumulus oophorus and zona pellucida and discus proliferus. So as I said, I'll discuss every diagram. So I'm discussing every diagram. So in a diagram to show the intimate relationship of oocyte and follicular cells, gap junctions are present between the microvilli of two. So you can see this is oocyte and follicular cells. There are gap junctions between zona pellucida, follicular cells. And this is the perivitelline space, polar body, zona pellucida, not present only in follicular cells. Follicular cells and its microvillus of oocyte. So how they are forming gap junction. So you can see relationship of a growing ovarian follicle to the ovary. Epithelial lining, follicle, ovary. As follicle is keep on increasing, you can see the stroma and theca become very thin here, and the ruptured follicle, ovum. These are fimbriae. We know we're trying to fallopian tip. Take up this. So structure of woman at the time of ovulation will be like this. The first polar body, spindle for second meiotic division, perivitelline space, cells of corona radiata, that is Cumulus oophorus cells are arranged and forming corona radiata, zona pellucida, witless cytoplasm. And the transformation of follicular cells to luteal cells. So granular cells in the wall of follicle collapse, become folded cells hypertrophy to form corpus luteum. So we know corpus luteum, when the ovulation takes place, the remaining yellow part is the corpus luteum. Next forms small cells in the wall of follicle soon after a rupture, considerably enlarged cells of corpus lutein pigment blood vessel. So fate of ovarian follicles, primordial follicles that do not rupture, degenerate, and the ovum and granular cells disappear. One follicle matures and sheds an ovum. The ruptured follicle becomes a corpus luteum degenerates and becomes corpus albicans. The cells of the interna persist as interstitial gland. Interstitial gland degenerates and becomes a corpus albicans. So here we have completed all the we have. These are like effects of non-destruction during first meiotic division. So you can see these are like empty. So finally they're forming trisomy here, that is a deformity here. And here also they're forming trisomy and also monosomy. So fertilization with normal gametes contain 23. So these are deformities. So yeah, we have completed all the diagrams. So here what is a deformity, let us understand. So first meiotic division 46 into 24 and 22. Yeah, actually they should divide equally, right, into 23, but one more is added extra over here, so this has become empty. So the cell cycle continues here empty and this is like extra, so that it is forming trisomy. So the same thing is also taking place here for you. Yeah, here first thing is normal like 23, but here in the second meiotic division, here the problem arises. Here at the first meiotic division only the problem arises. So as here in the second this is empty going on, so that again it is forming trisomy and monosomy. So that's defects. So the effects of non-distension in first meiotic division, this is the effects of non-distension in the second meiotic division. So by this we completed all the pictures of both first and second lesson and also completed all the theory part.